Immunological Tolerance and Autoimmunity. This is our discussion for the book of Cellular and Molecular Immunology by Abbas and colleagues. And this is chapter 15 in the book. If you are following, just have your book and let us read together this chapter. So here we will discuss overview of immunological tolerance, T lymphocyte tolerance, central T cell tolerance, peripheral T cell tolerance, factor that determine the tolerogenicity of self-antigen, B lymphocyte tolerance, central B cell tolerance, peripheral B cell tolerance, tolerance to conventional microbes and other foreign antigen, therapeutic tolerance, induction, mechanism of autoimmunity, general features of autoimmune disorder, immunological abnormality leading to autoimmunity, genetic basis of autoimmune, role of infection in autoimmune, and other factors in autoimmunes. Then you will see the summary. So, immunological tolerance is defined as unresponsiveness to an antigen that is induced by exposure to that antigen. The term arose from the experimental observation that animal that had encountered an antigen under a particular condition would not respond to, that is, would tolerate subsequent exposure to the same antigen. When specific lymphocytes encounter antigen, the lymphocyte may be activated, leading to immune response, or the cells may be inactivated or eliminated, leading to tolerance. The same antigen may induce an immune response or tolerance depending on the condition of exposure and the presence or absence of other concomitant stimuli such as co-stimulators. Antigens that induce tolerance are called tolerogens or tolerogenic antigen, to distinguish them from the immunogen, which generate immunity. Tolerance to self-antigen, also called self-tolerance, is a fundamental property of the normal immune system, and failure of self-tolerance results in immune reactions against self or autologous antigen. Such reactions are called autoimmunity, and the disease they cause are called autoimmune disease in chapter 1. In chapter 1, we discussed we introduced the concept of self-non-self discrimination, which is the ability of the immune system to recognize and respond to foreign antigens, but not to self-antigen. McFarlane Burnett, among the first hypothesized clone selection, added the corollary that lymphocytes specific for self-antigen are eliminated to prevent immune reaction against one's own tissue. We now know there are multiple mechanisms of self-tolerance in addition to elimination of self-reactive lymphocytes. Elucidating these mechanisms and why they sometimes fail is the key to understanding the pathogenesis of autoimmunity. In this chapter, we will discuss the immunological tolerance mainly in the context of self-tolerance and how self-tolerance may fail, result in autoimmunity. We will also consider the tolerance to foreign antigens and potential of tolerance induction as therapeutic strategies for allergic and autoimmune disease. Induction of tolerance to prevent the rejection of the cell and organ transplant is discussed in chapter 17. Overview of immunological tolerance. There are several characteristics of tolerance in T and B lymphocyte population. It is important to appreciate the general principle before we discuss the specific mechanism of tolerance in these lymphocytes. The mechanism of tolerance eliminates and inactivates lymphocyte that expresses high affinity receptor for self-antigen. All individuals inherit essentially the same antigen receptor gene segments, and these recombine and are expressed in lymphocyte as cells arise from the precursor cell. The specificities of the receptor encoded by the recombined genes are random and are not influenced by what is foreign or self for each individual. It is not surprising that during this process of generating a larger and diverse repertoire, some developing T and B cell in every individual may express a receptor capable of recognizing normal molecule in in that individual. This is self-antigen. Therefore, there is a risk for lymphocyte to react against that individual cell and tissue, causing the disease. The mechanism of immunological tolerance prevents such a reaction. Tolerance is antigen-specific, resulting from a recognition of antigen by individual clones of lymphocyte. This contrasts with the therapeutic immunosuppression, which affects lymphocyte of many specificities. The key advances that allowed immunologists to study tolerance were the ability to induce this phenomena in animal by exposure to defined antigen under various conditions, and to then analyze the survival and function of lymphocytes that had encountered the antigen. In 1950s, Peter Medrow and colleagues showed that neonatal mice of one strain exposed to cell from other strain become unresponsive to subsequent skin graft from donor strain. Later studies show that tolerance could be induced not to foreign cell but also to protein and other antigen. Any antigen may be an immunogen or tolerogen, depending on the numerous factors such as antigen exposure during lymphocyte maturation and recognition by specific lymphocyte in the presence or absence of innate immune response. These factors are discussed later in the chapter. Self-tolerance may be induced in immature self-reactive lymphocytes in the generative lymphoid organs, central tolerance, or in mature lymphocyte in the peripheral site, that is called peripheral tolerance. A central tolerance ensures that the repertoire of mature naive lymphocytes become incapable of responding to self-antigen that are exposed in the generative lymphoid organs, the thymus for T cell and the bone marrow for B cell, also primarily or central lymphoid organs. However, central tolerance is not perfect, and many self-reactive lymphocytes complete their maturation and are present in healthy individuals. Therefore, mechanisms of peripheral tolerance are needed to prevent activation of these potentially dangerous lymphocytes. You have the diagram. This is the central and peripheral tolerance to self-antigen. In central tolerance, immature lymphocytes specific for self-antigen may encounter this antigen in the generative lymphoid organs, referred to the central organ in the context of tolerance induction, and are deleted, change their specificity B cell only, or in case of CD4 T cells, develop into regular lymphocyte Treg. In peripheral tolerance, some self-reactive lymphocyte may mature and enter peripheral tissue and may be inactivated or deleted by an encounter with self-antigen in these tissues and are suppressed by regulatory T cell, Treg, peripheral tolerance. So this is the diagram that we see. We have lymphoid precursor, immature lymphocytes, then strong recognition of self-antigen leading to apoptosis, or it can lead to change in a receptor editing in B cells, or development of regulatory T lymphocyte CDT cell only. That's going to occur here. So, or sometimes if weak or no recognition of self-antigen, they mature. So this is the path gate of what is happening actually in the body. Lymphocytes are produced. When they are produced, they are immature. They are tested. They are tested with antigens. This is antigen presenting cell. If they capture or if they attach strongly, then they either undergo apoptosis, they change in receptor or receptor editing B cell. For B cell, they can edit, change their character, and some of them may development of regulatory T lymphocyte, CD4 T cells can happen. The CD4 T cells and those which will be edited and agree with the B cell, they can pass this stage, but those which cannot, they will undergo apoptosis and deletion. But those which will react weak on recognition of self-antigen, then they mature. Then the recognition of self-antigen, again, this is the secondary in the peripheral. In the peripheral, this will happen in recognition of self-antigen. They can undergo apoptosis or they can undergo anage, or sometimes they can be regulated by what? Can be suppressed by T regulatory T cell. This is what is happening actually. Recognition of self-antigen in peripheral. And anage is very common, suppression is very common, and apoptosis as well. Central tolerance occurs during a stage in maturation of lymphocyte when when an encounter with an antigen may lead to the cell death or replacement of self-reactive antigen receptor with one that is not self-reactive. As lymphocytes are maturing in the generative lymphoid organs, immature cells may encounter antigen in these organs. The antigen that are present in these organs are mostly self-antigen because the antigen that are present in these organs are mostly self-antigen because foreign, e.g. microbial antigen that enter from the external environment are typically captured and taken to secondary lymphoid organs such as the lymph node, spleen, mucosa, lymphoid tissue, and are not concentrated in thymus or bone marrow. The antigen normally present in the thymus and bone marrow include ubiquitins, widely disseminated self-antigen, some of which may be expressed by cells in thymus and other may be brought in by the blood. In addition, many peripheral tissue-specific antigen are expressed in thymus by a special mechanism that is described later. Therefore, in the generative lymphoid organ, the immature lymphocytes that recognize antigen are typically cells with high affinity receptor specific for self-antigen. The fates of immature lymphocytes that recognize self-antigen with high affinity are described later. Mature lymphocytes that recognize self-antigen in peripheral tissue become incapable of activation by exposure to antigen or die by apoptosis. These mechanisms of peripheral tolerance are important for maintaining and responsiveness to self-antigen that are expressed in peripheral tissue and are not in generative lymphoid organ, and for tolerance to antigen that are expressed after mature lymphocytes specific for these antigen have already been generated. Peripheral tolerance is also maintained by regulatory T cell, Treg, that actively suppress the activation of lymphocytes specific for self and other antigen. Treg mediated suppression occur in secondary lymphoid organs and non-lymphoid tissue. In addition to suppressing the immune response to self-antigen in peripheral tissue, Treg may also prevent response to antigen derived from commensal microbes or from the fetus in a pregnant female. Some self-antigen are sequenced from immune system and other antigen are ignored. Antigen may be sequenced from the immune system by anatomical barrier, such as in testes and eyes, and thus cannot engage antigen receptor. Commensal microbes also may be separated from the immune system by physical barriers such as keratin layers of skin and mucus layer of gastrointestinal and the respiratory tract. In experimental model, some self-antigen are available for recognition by lymphocyte, but for unknown reason fail to elicit any response and other functionally ignored. The importance of this phenomena of ignorance for maintenance of self-tolerance is not established. The induction of immunological tolerance is a possible therapeutic strategy for preventing harmful immune response in autoimmune and allergic disease and transplant rejection. Tolerance induction also may be useful for preventing immune reaction to the product of newly expressed gene in gene therapy protocol for preventing reaction to injection protein in patient with the deficiency of these proteins or patient with hemophilia treated with factor VIII and for promoting acceptance of stem cell transplant. Experimental approach, especially the creation of genetically modified mice, have provided a valuable model for analysis of self-tolerance, and many of our current concepts are based on studies with such models. Furthermore, by identifying mutation and genetic polymorphism that may be associated with autoimmune in mice and human, it has been possible to deduce some of the mechanism of self-tolerance. However, for most self-antigens, we do not know which one induce central or peripheral tolerance, or rare ignored. More importantly, it is also not known which tolerance mechanism fail in common human autoimmune diseases, and this remains a major challenge in understanding autoimmunity. We will discuss central and peripheral tolerance first in the T cell and then in B lymphocyte, but many aspects of the process are common to both lineage. T cell tolerance, T lymphocyte tolerance. Much of our understanding of tolerance to self-antigen is based on studying this process in T lymphocytes. This is in part because immunologists have developed informative experimental model for studying T cell tolerance. In addition, many of the therapeutic strategies that have been developed to induce tolerance to transplant and autoantigen are aimed at inactivating or eliminating T cell. This is because pathologic inflammation reactions are typically mediated by T cell, and CD4 plus helper T cell also promote the production of potentially injury antibody by B cells. Central T cell tolerance. During their maturation in thymus, many immature T cells that recognize antigen with higher avidity die, and some of the surviving cells in the CD4 lineage develop into Treg. Death of immature T cells as a result of recognition of antigen in thymus is known as deletion or negative selection. It was described in chapter 8 in the discussion of T cell maturation. This process affects class 1 and class 2 major histoconvertibility complex restricted T cells and therefore important for tolerance in both CD4 and CD8 lymphocyte population. Negative selection of thymocytes is responsible for the fact that the repertoire of mature T cells that leave the thymus and populate peripheral lymphoid tissue is unresponsive to many self-antigens that are present in the thymus. Negative selection occurs in double positive T cell in thymic cortex and newly generated single positive T cell in medulla. In both locations, immature thymocytes with high affinity receptor for self-antigen that encounter this antigen die by apoptosis. The two main factors that determine if a particular self-antigen will induce negative selection of self-reactive thymocytes are the presence of that antigen in thymus by local expression or delivery by blood, and affinity of the thymus T cell receptor that recognize the antigen. Thus, important questions that are relevant to negative selection are which self-antigen are present in thymus and how immature T cell that recognize this antigen are deleted. That remain big questions. So we have the diagram here. The diagram is central T cells tolerance, recognition of self-antigen by immature T cells in thymus lead to the death of the cells, negative selection, deletion, or to the development of regulatory T that enters the peripheral tissue. So we have the negative selection, deletion, we have the thymus, development of regulatory T cells. So what happened is in thymus, immature T cell specific self-antigen is presented. If it reacts too strongly, it dies. But some of them which they react strongly, they can change into regulatory T cells. So they go to the peripheral, and these are the ones responsible now to control for homeostasis of T helper 1 and T helper 2 together with the other states. The antigens that are present in thymus include many circulating and cell-associated proteins that are widely distributed in most tissues. The thymus also has a special mechanism of expressing numerous protein antigens that are not ubiquitously expressed, but rather are limited to certain peripheral tissues, so that immature T cell specific for these antigen can be deleted from the development from the developing T cell repertoire. These peripheral tissue antigens are produced in medullary thermic epithelial cells under the control of autoimmune regulator proteins. Mutation in autoimmune regulator genes are the cause of autoimmune disease called autoimmune polyendocrine syndrome type 1, APS1. This group of disease is characterized by antibody and lymphocyte-mediated injury to multiple endocrine organs, including the parathyroid, adrenal, and pancreatic isolates, as well as skin and other tissues. A mouse model for autoimmune polyendocrine syndrome type 1 has been developed by knockout of AR genes and it recapitulates many of the features of human disease. Studies with mice have shown that several proteins that are produced in the peripheral organs, such as pancreatic insulin, are also expressed at low level in MTAC by the AR-dependent mechanism, and immature T cells that recognize these antigens are deleted in thymus and or the T cell develop into Treg. In the absence of functional error, as in APS1 patient and R knockout mice, these antigens are not expressed in thymus, and CD4 and CD8 T cells, specifically for antigen escape deletion, mature and enter the peripheral where they attack the target tissue in which the antigen are expressed independent of error. Patients also make a variety of autoantibodies, probably because of failure to delete helper T cells specific for self-antigen. Interestingly, some of these autoantibodies react against and neutralize their own interleukin 17, interleukin 22, and type 1 interferons. The deficiency of interleukin 17 and perhaps interleukin 22 makes these patients susceptible to mucocutaneous candidiasis, which is common features of APS1 and reflect the essential role of T17 cytokine in defense against this fungal infection, already seen in chapter 10. The IRA protein may function as transcriptional regulator to promote the expression of selected tissue-restricted antigens in the thymus. It is a component of multiproteins complex that is expressed mainly in MTACs and involved in transcription elongation and chromatin remodeling. However, drive expression of wide range of tissue antigen and not other protein is still not known. AERA is also expressed in the peripheral tissue, but its role in elimination of self-reactive T cells in the peripheral is not established. High affinity T cell receptor signaling in immature T cells trigger the mitochondrial pathway for apoptosis. The mechanism of apoptosis are described later in this chapter when we discuss deletion and the mechanism of peripheral T cell tolerance. Clearly, immature and mature lymphocytes interpret antigen receptor signal differently. The former die and later are activated. The biochemical basis of difference is not known. Some self-reactive CD4 plus T cells that see the self-antigen in thymus are not deleted but instead differentiate into Treg that are specific for these antigen. See in figure 15.2 already seen. The regulatory cells leave the thymus and inhibit response against self-antigen in peripheral. What determines the choice between deletion and development of Treg is not known. Possible factors include affinity of antigen recognition, the types of antigen presenting cells presenting the antigen, and the availability of certain cytokines locally in the thymus. We will describe the characteristics and function of Treg later in the context of peripheral tolerance because these cells suppress immune response in peripheral. Peripheral T cells tolerance. The mechanism of peripheral tolerance are anage, functional and responsiveness, suppression by Treg, and deletion, cell death. This is seen in figure 15.4. This mechanism may be responsible for T cell tolerance to tissue-specific self-antigen, especially those that are not abundant in thymus. We do not know if tolerance to different self-antigen is maintained by one or another mechanism, or if all of these mechanisms function cooperatively to prevent autoimmunity. The same mechanism may also induce unresponsiveness to foreign antigens that are presented to immune system under tolerogenic condition. So here we have the function of R in deletion of T cells in thymus. A is the autoimmune regulator R. Autoimmune regulator protein is that part of the complex that regulates the expression of tissue-restricted antigen in medullary thermic epithelial cells, MTEC. Other proteins in this transcription regulator complex are not shown. Peptides derived from this antigen are displayed on the MTEC, the medullary thermic epithelial cells, and recognized by immature antigen-specific T cell, leading to the deletion of many self-reactive T cells. So, on A cell, we say we have tissue-restricted antigen, TRAS, R, which is autoimmune regulator, we have here. These medullary epithelial cells, so if you are having immature reactive T cells, if it reacts, this one, if when it reacts, it can form either Treg development or it can form negative selection deletion. On this aspect, in the absence of functional error, these immature self-reactive T cells are not eliminated. They can mature into naive T cell that can be activated in secondary lymphoid organ, generating effector T cells not shown that enter tissue where the antigen continue to be produced and cause injury, although only CD4 T cells are shown. R also control the deletion of CD8 T cells. So what happened? You don't have R. This cell is there immature. Nothing is controlling it, so they cannot undergo apoptosis. They mature into reactive T cells, and this one failure of negative selection, and therefore they can cause a lot of autoimmune diseases. ANAGE, functional unresponsiveness. Exposure of CD4 T cells to an antigen in the absence of co-stimulation or innate immunity may make the cell incapable of responding to that antigen. In this process, which is called ANAGE, the self-reactive cells are not killed, but they become unresponsive to antigen. We previously introduced the concept that full activation of T cells require the recognition of antigen by T cell receptor, which provide a signal 1. The recognition of costimulatory mainly B7-1 and B7-2 by CD28 signal 2. Prolonged signal 1, antigen recognition alone may lead to the ANAGE. It is likely that self-antigen are continuously displayed to specific T cell in absence of innate immunity and strong co-stimulation. Antigen-induced energy has been demonstrated in the variety of experimental models, including studies with T cell clones exposed to the antigen in vitro, which were the basis for the original definition of energy. And it is experiment in which antigen are administered to mice without adjuvant. Other studies have used the transgenic mice in which particular protein antigen are expressed throughout life and recognized by T cell in absence of inflammation or innate immune response that normally accompany exposure to microbes. There is evidence that energy is the mechanism of tolerance to some self-antigen in humans as well. Energy cells may survive for days or weeks in quiescent state and then die. Several mechanisms may function to induce and maintain anergic state. T-cell induced signals transduction is blocked in energy cell. The mechanism of this signaling block are not full and known. In different experimental model, it's attributable to decreased T-cell receptor expression, perhaps because of increased degradation, see later, and the recruitment to T-cell receptor complex of inhibitory molecules such as tyrosine phosphatase. Self-antigen recognition without co-stimulation may activate cellular ubiquitin ligase, which ubiquitinate T-cell receptor-associated protein and target them for proteolytic degradation in the proteasomes or lysosomes. The net result is loss of these signals molecule and the defective T-cell activation. One ubiquitin ligase that is important in T-cell is CBLB. It's already in chapter 7. Mice in which genes encoding CBLB is knocked out shows spontaneous T-cell proliferation and manifestation of autoimmunity, suggesting that this enzyme is involved in maintaining T-cell and responsiveness to self-antigen. It is not known why self-antigen recognition, which occur typically without strong co-stimulation, activate these ubiquitin ligase, whereas foreign antigen that are recognized with co-stimulation do so much less or not at all. So we have the diagram here, the mechanism of peripheral T-cell tolerance, signal involved in the normal immune response, and three major mechanisms of peripheral T-cell tolerance. So we see here, normal dendritic cell is in interaction with, this is normal effector memory cell, then anage, then functional and responsiveness, suppression, block activation because of regulatory. So regulatory T cell, it can block the activation, and when it blocks the activation, now everything's happened. Now the cell is not activated and therefore no effect is happening. And these apoptosis can happen during deletion. When T-cell recognize self-antigen in the absence of innate immune response, they may engage inhibitory receptor of CD28 family. Those function is to terminate the T-cell response. The function of the best known inhibitory receptor of T-cell are described in the following section. Regulation of T-cell response by inhibitory response. In chapter 9, we introduced the general concept that outcome of antigen recognition by T-cell is determined by the balance between engagement of activating and inhibitory receptor at the time of T-cell receptor recognition of antigen. Although many inhibitory receptors have been described, the two whose physiological role in self-tolerance is best established are CTLA-4 and PD-1. These receptors have been called co-inhibitor. To contrast them with co-stimulator, studies of these inhibitory receptors have increased our understanding of tolerance mechanism and led to new therapeutic approach for manipulating immune responses. CTLA-4 cytotoxic T lymphocyte antigen 4, so named because how it was discovered, is a member of CD28 receptor family, see in figure 9.5, and like the activating receptor CD28, it binds to B7 molecules. The importance of CTLA-4 in tolerance induction is illustrated by finding that knockout mice lacking CTLA-4 and people with loss of function mutation in CTL genes, discussed in more detail below, develop inflammatory lesions containing activated T cells and macrophage affecting multiple organs. These results indicate that defect in these one control mechanism result in failure of self-tolerance, Polymorphism in the CTLA4 genes are also associated with several autoimmune diseases in humans, including type 1 diabetes and Graves' disease. CTL4, which is cytotoxic T lymphocyte antigen 4, function as competitor inhibitor of CD28 and reduce availability of B7 for CD28 receptor. CTL has an unusual mechanism of action. It is expressed constitutively at high level on Treg and transiently on recently activated T cell, and it prevents the activation of responding T cells. In other words, CTLA4 on one T cell, Treg, can inhibit response of other T cells. Recall that CD28 and CTLA recognize the same ligands B71, which is equivalent to CD80, and B72, which is equivalent to CD86. CTLA4 has 10 to 20-fold higher affinity for B7 than does CD28. The cytoplasmic tail for CTLA4 does not appear to have any significant function. Instead, it contains a motif that connects it to the clathrin, a protein involved in receptor-mediated endocytosis. Because of this, CTLA4 is an endocytotic receptor that binds to B7 molecule on antigen-presenting cell and remove and ingest these molecules, a process that has been called transendosis, in which one cell endocytosis or endocytes a protein from another cell. Therefore, when CTL4 is expressed on either Treg or activated T cell, it outcompetes the CD28 and reduces the amount of B7 available on antigen-presenting cell to provide co-stimulation via CD28. This competitor inhibitor is especially important when B7 level on antigen-presenting are low, as on resting antigen-presenting cell displaying self-antigen. When B7 level increases, for example, after exposure to microbes, there is relatively more engagement of low affinity receptor CD28 because CTLA limits the initial co-stimulation dependent activation of T cell in secondary lymphoid organ. Mutating or blocking this receptor leads to severely dysregulated immune response with enlarged lymph nodes, lymphoproliferation, and multi-organ inflammation. So let us see the diagram. Mechanism of T cell energy. This is mechanism of T cell energy. This is for energy actually, not for CTS4, for CTL4. So a T cell's response are induced when the cell recognizes an antigen presented by proliferation of antigen presenting cell and activating receptors on the T cell such as CD28 recognize co-stimulator on the antigen presenting cells such as B7. If the T cells recognize a self-antigen without co-stimulation, the T cell will read there. Then here is normal. Antigen recognition, DC expression co-stimulator, naive T cell activating signals comes, recognition of foreign antigen happen. Then we have T cell response, effector T cells, T cell proliferation, and differentiation. On T cell energy, what happens is dendritic cell present. This is the naive T cell recognition of antigen. It can either go to signaling block and no response, or it can happen whereby inhibitory receptor, engagement of inhibitory receptor in that prevent the activation. Then that is done. So the T cells become unresponsive to antigen because of blocking in signaling from T cell receptor complex and engagement of inhibitory receptors such as cytotoxic T lymphocyte antigen. And prolonged cell death proteins and signaling block may be result of recruitment of phosphatase to T cell receptor complex or activation of ubiquitin ligands that degrade signal proteins. T cell remain viable, but it is unable to respond to self-antigen. that is dendritic cells and other things. Mutation of even one allele of CTL4 causes systemic inflammation involving multiple organs with severe lymphoproliferation. It is thought that haploinsufficient reduce the level of expressed CTL4 enough that it cannot compete effectively with CD28. Mutation affecting a protein called LRBA that is involved in the endocytosis and recycling for CTL4 cause a similar systemic inflammatory disease, further emphasizing the critical role of CTL4 trafficking in its inhibitory function. Because these diseases are caused by excessive BL-CD28 mediated co-stimulation resulting from failure of CTL4 mediated competition, they can be treated by blocking B7 molecule with the drug CTL4-IgG. The realization that CTL4 limit and act on a checkpoint in immune response has led to the idea that lymphocyte activation can be promoted by reducing inhibition. Blocking CTL4 with an antibody results in increasing immune response to tumor, a process known as checkpoint blockade. You see it in chapter 8. And CTL4 antibody is now approved for treatment of advanced melanomas and other cancers. Predictively, one of the treated patients develop a manifestation of autoimmune with the inflammation in various organs. PD1. Another inhibitory receptor of the CD28 family is PD1, programmed cell death protein 1, so called because it was originally thought to be involved in programmed cell death, but now known to have role in T-cell apoptosis. PD1 recognizes two ligands called PD1, PDL1, PDL2. PDL1 is expressed on antigen-presenting cell and many other tissue cells. PDL2 is expressed mainly on the bone marrow-derived antigen-presenting cells. The receptor for PD1 is expressed on antigen-activated T cells. Engagement of PD1 by either of its ligands leads to the phosphorylation of immunoreceptor tyrosine-based inhibitory motif and switch motif in cytoplasmic tails. See chapter 7. The motif bind the phosphatase SHP2, which remove phosphatase from various substrates. Thus, PD1 counteract kinase-dependent signal from T-cell receptor, co-receptor complex, and from the CD28 and other costimulatory receptor, resulting in inactivation of T cells. Mice in which PD1 is knocked out develop autoimmune that is typically milder than that seen in CTLA4 knockout. PD1 expression on T cells increases with antigen stimulation, so it is especially important for controlling response to prolonged antigen exposure, as with self-antigen, tumor, chronic infection. Checkpoint blockade with anti-PD1 and anti-PDL1 antibodies used to treat wide variety of cancer, but it's also associated with the development of autoimmune reaction. We will see it in chapter 18. So, mechanism of action of CTL4, a cytotoxic T lymphocyte antigen expressed on regulatory T cell or activated, can inhibit. So we are told that a cytotoxic T lymphocyte antigen 4 expressed on regulatory T cells or activated T cell can inhibit the activation of responding T cells on the same antigen presenting cells. So here we see we have CTL expressed the T cell reg. So this one, if it expresses it, we have the antigen presenting cell, it is now blocked. Block, remove BDL, lack of co-stimulation, T cell and responsiveness, then there is no responsiveness. B, this is for B, sorry, but it's C cell. This is for A. and for B is CTL. And for this CTL on regulatory or activated T cell bind to B cell molecule on antigen presenting and remove this molecule from surface of antigen presenting cell, making the B cell co-stimulator unavailable to CD28 and blocking T cell activation. This action of CTL4 is able to suppress immune response best when the B7 level are low, enabling the CTL to outcompete the low affinity receptor CD28. Although both CTLA4 and PD1 establish checkpoint in immune response, their role may be complementary and not identical. For example, PD1 appear to be most important for terminating the response of effector T cells, especially CD8 cells in peripheral tissue, whereas CTLA4, as discussed previously, limit the initiation activation of T cells in secondary lymphoid organs. Although the principal function of CTLA4 is to prevent response to self-antigens, but TD1 may have evolved to limit excessive reaction against microbes, especially viruses, and reduce immunopathology associated with these infections. Those for their major, some of their major differences are summarized in table 15.1. So this is the mechanism of action of PD1 engagement of its ligands. Most PD1 PDL1 by PD1 programmed cell death protein 1 on T cells recruit a receptor associated with phosphatase SHP2 to an item and ETSM in cytoplasmic tail of PD1s, which remove phosphatase from other protein and inhibit kinase dependent signal from CD28 and T cells receptor complex. Note that only some signal from a T cell receptor complex and CD28 are shown. Antigen presenting cells and item is immunoreceptor tyrosine based activation motif. Item is immunoreceptor tyrosine based inhibitory motifs. And ITSM is immunoreceptor tyrosine based switch motif. PD1 is programmed cell death protein 1. TCR is T cell receptor. So you see here, and we have antigen presenting cell. Normally it causes activation, but in the presence of PD1 ligand here, when you have the PD1, this PD1 can bind and can compete with as normal with CD20. Then when this want to be activated, it will be blocked. So mechanism of blockage is just around here. So when it is blocked, the PD1 inhibit signal from total cell receptor complex and CD28. So action and function of CTL and PD1. Major site of action, secondary lymphoid organs. For CTL4 is secondary lymphoid organ. PD1 is peripheral tissue. Stage of immune response that is inhibited, induction, priming. Here effector phase. Here induction. Cell that is inhibited, CD4 more inhibited than CD8. Here in this part of PD1, CD8 is more inhibited than CD4. Main cellular expression, it is expressed, the CTL4 is expressed mostly in a Treg and activated T cell, and the PD1 is expressed in CD in activated T cells. Main signal inhibited, competitive inhibitor for CD28 co-stimulation by binding to B7 with high affinity and removing B7 from antigen presenting cell. Here signaling inhibitor of CD28 and T cell receptor inhibit kinase dependent signal from CD28 and the T cell receptor by recruiting the SHP2. Principal physiologic roles postulated. preventing autoimmune response to self-antigen. This is preventing excessive pathological response to microbes, especially viruses and some antigens. Several other inhibitory receptors have been identified, including some belonging to tumor necrosis factor receptor family and other T-cell immunoglobulin and mucin TIM family. The significance and biological roles of these receptors are not well established, as the mechanism of action and function of CTL4 and PD1. There is a great interest in defining the role of these receptors in regulation of immune response and in targeting these molecules therapeutically, especially for cancer immunotherapy. Suppression by regulatory T cells. The concept that some lymphocyte could control the response of other lymphocytes was proposed many years ago and was soon followed by experimental demonstration of a population of T lymphocytes that suppressed immune response. These initial findings led to the enormous interest in suppressor T cell, which became one of the dominant topic of immunology research in 1980s. However, this field has had a somewhat checkered history, mainly because attempts to define population of suppressor cells and their mechanism action were largely unsuccessful. In 1990s, the idea had an impressive rebirth with the application of better approaches to define, purify, and analyze the population of T lymphocytes that inhibit immune response. These cells are called regulatory T cells. Regulatory T cells are a subset of CD4 T cell, whose principal function is to suppress immune response. See figure 15.8. CD4 Treg expresses high level of interleukin-2 receptor alpha chain CD25 and the transcription factor called FOXP. FOXP3 is the member of Foxhead family of transcription factor that and is critical for development of function of the most Treg. The essential role of FOXP3 Treg in maintaining self-tolerance and preventing autoimmunity have been established by analysis of inherited diseases. A rare systemic autoimmune disease in humans called IPEX, immune dysregulation polyendocrinopathy enteropathy X-linked syndrome, is caused by loss of function of mutation in FOXP3 genes, which result in deficiency of Treg. This disease is fatal unless the children are given hematopoietic stem cells transplant, which restore normal Treg. Mice with spontaneous or experimentally induced mutation in FOXP3 gene develop similar multisystem autoimmune disease associated with the absence of CD25 Treg. And the disease can be prevented by introducing normal Treg. This observation has established the importance of Treg for maintaining self-tolerance. Recent surge of interest in Treg is because of an increasing appreciation of their physiological roles, as well as possibility that defect in these cells may result in various autoimmune disease, and conversely that Treg can be administered or expanded to treat inflammatory disease. Phenotypic markers and heterogeneity of regulatory T cell. Although numerous T cell populations have been described as possessing a suppressive activities, the cells type whose regulatory role is best established is CD4, FOXP3, CD25 high, FOXP3, and CD25 are essential for generation, maintenance, and function of this cell. This cell usually expresses a low level of receptor for interleukin 7, CD2127, and are predicted from this pattern of receptor expression. They use interleukin 2, but not interleukin 7 as their growth and survival factor. FOXP3 Treg typically expresses high level of CTL4, which is also required for their function. Demethylation of FOX genes locus of the lock-containing genes that are expressed in these cells serve to maintain a stable regulatory T cell phenotype, And these epigenetic changes are now used to identify Treg in the basic and clinical research. FOXP3 Treg are classified as thermic or peripheral based on where they develop, discussed next. They also have subset of Treg that are distinguished by history of antigen response, naive, activated, and memory, or tissue residence, lymphoid and non-lymphoid. Treg that are located in peripheral tissue, such as intestine, lung, skin, and fat, may have functions that are unique to each tissue, discussed later. Treg that have been described in the follicle of secondary lymphoid organs, where they may inhibit antibody response. Treg have been described in the follicle of secondary lymphoid organs, where they may inhibit antibody response. In different types of immune response, Treg may be induced to express the same chemokines receptor as effector T cells. This is one way in which Treg colonize in tissue with effector cell, enabling the Treg to control diverse immune reaction in tissue. Generation and maintenance of regulatory T cell. Treg are generated mainly by self-antigen recognition in thymus and also by recognition of self and foreign antigen in peripheral tissue. In thymus, development of Treg is one of the fates of T cells committed to CD4 lineage that recognize self-antigen. In peripheral tissue, antigen recognition in the absence of strong innate immune response favor the generation of regulatory T cells from naive T cell lymphocytes. Treg can also develop after inflammatory reactions. The majority of Treg in lymphoid tissue are thought to be derived from thymus as consequence of self-antigen expression, although many markers have been proposed to distinguish thymic from peripheral Treg. Whether these markers can be used to reliably distinguish the two subsets in lymphoid organ or blood of mice and human has not been established. The generation, survival, and functional competence of Treg are dependent on cytokine interleukin-2. Patients with homozygous mutation in interleukin-2 receptor alpha and beta chain develop a systemic autoimmunity and have few functional Treg. Mice in which the genes of interleukin-2 for alpha and beta chains of interleukin-2 receptor is knocked out also develop autoimmune manifestation of inflammatory bowel disease. Autoimmune hemolytic anemia and multiple autoantibody including anti-erythrocytes and anti-DNA antibodies could as well occur. These mice have greatly reduced number of CD25 FOXP3 Treg, and their disease can be corrected by injecting these cells. Interleukin-2 promote differentiation of T cells in thymus into the regulatory subset and is required for maintenance of this cell population in the peripheral. Because FOXP3 Tregs do not produce interleukin-2, this growth factor is provided by conventional T cells responding to self or foreign antigen. Interleukin-2 activates the transcription factor STAT5, which may enhance expression of FOXP3 as well as other genes that are involved in the functioning of Treg. These results are the basis for ongoing clinical trial testing the ability of interleukin-2 to promote Tregs in the human for the control of graft versus host disease, autoimmune inflammation, and graft rejection. The generation of some Treg requires cytokine transforming growth factor. Culture of naive T cells with the activating anti-T cell receptor antibody together with transforming growth factor beta and interleukin-2 discussed earlier can induce the development of regulatory cells in vitro. In mice, elimination of T transforming growth factor beta or blocking the transforming growth factor in the T cell lead to systemic inflammatory disease because of uncontrolled leukocyte activation. Transforming growth factor stimulate expression of FOX, which could be the major roles in T-reg development. So we have normally the regulatory T cells. This is what the diagram for. See the figure 15.8. Regulatory T cells are generated by self-antigen recognition in the thymus, sometimes called natural regulatory cells, and probably to the lesser extent by antigen recognition in peripheral lymphoid organ called inducible or adaptive regulatory cells. The development and survival of these Treg require interleukin-2 and transcription factor FOX P3 in the peripheral tissue. Treg suppresses activation of effector function of other self-reactive and potentially pathogenic lymphocytes. The dendritic cell, there's an explanation. So we have the thymus. In here, the recognition of self-antigen, it can change it to the FOX P3. Then the FOX P3 is resulting formation of a regulatory T cell. These regulatory T cells, they can they function inhibition of T cell response. So they function to inhibit the naive T cells and other things, inhibition of the other cells. So in peripheral, this recognition of antigen in peripheral tissue, it can again cause the FOX. Mostly this again happen, I think, in other diseases like mostly the parasite. We have another diagram here. 19, role of interleukin-2 in maintenance of regulatory T cells. Interleukin-2 produced by conventional T cells responding to self or foreign antigen acts on regulatory T cells. Recognize antigen on antigen presenting dendritic cell and promote the survival and function of Treg, enabling them to control response of conventional T cells. See the good part, eh? The dendritic cell, regulatory T cell, self-foreign antigen, then this result to the this is antigen, then it presenting it again, it releases the interleukin 2. Interleukin 2 lead more activation of Treg. Then we have this again, the dendritic cell, but with a lot of proliferated what? Treg. Then they control this one, then it's not activated. So activation of this one, if it continues as it continues, the more activation is happening, the more T cells are produced, and later on they can cause the body not to continue to be activated. And this process, if it is high, then it can inhibit the whole process. Mechanism of action of regulatory T cells. Treg appear to suppress immune response at multiple steps, at the induction of naive T cell activation in lymphoid organ, as well as effector phase of this response in tissue. They control T cell response mainly by inhibiting the stimulatory ability of dendritic cells and also by suppressing T cell activation. Treg may also suppress B cell activation and inhibit proliferation and differentiation of natural killer cells. Although numerous mechanisms of suppression have been proposed, the following are the best supported by the available data. Wow. The CTL4 mediated inhibition of co-stimulation. This is number 1. CTL4 on Treg bind to B7 molecules on antigen-presenting cells and remove these molecules, resulting in reduced availability of B7 for CD28 mediated co-stimulation. Production of immunosuppressive cytokine interleukin 10 and T growth factor beta. These cytokines are discussed below. Consumption of interleukin 2. Because of high level of suppression of interleukin 2 receptor, this cell may also absorb. Because of high level of expression of interleukin 2 receptor, this cell may also absorb interleukin 2 and deprive other cell population of this growth factor, resulting in a reduced proliferation and differentiation of interleukin 2 dependent cells. Wow. You're getting it, eh? It's good one. Whether all regulatory cells work by all these mechanisms or if there are a subpopulation that use different mechanisms to control immune response has not been established. In fact, there is some evidence in humans that two different populations of Treg can be distinguished by the expression of FOXP3 and production of interleukin-10, but this separate may not be absolute. Inhibitory cytokines produced by regulatory T cells. Transforming growth factor beta and interleukin-10 are involved in the generation and function of Treg. These cytokines are produced by and act on many other cell types in addition to regulatory T cells. Here we describe the properties and action of these cytokines. Transforming growth factor beta. Transforming growth factor beta was discovered as a protein made by tumor that promoted the survival of cancer in vitro. It is usually a family of closely related molecules encoded by distinct genes, commonly designed as transforming growth factor beta 1, transforming growth factor beta 2, transforming growth factor beta 3. Cells of the immune system synthesize mainly transforming growth factor beta 1. Transforming growth factor beta 1 is produced by CD4 Treg, activated macrophage, and other cell types. The processing of transforming growth factor beta, transforming growth beta receptor, and signal downstream of these receptors were described in chapter 7 already. So, transforming growth factor beta has many important and diverse roles in the immune system. A transforming growth factor beta inhibits the proliferation and effector function of T cells and the activation of macrophage. Transforming growth factor inhibits classical macrophage activation, but is one of the cytokines secreted by alternative activated macrophage, we have seen in chapter 10. Transforming growth factor beta also suppresses the activation of other cells such as neutrophil and endothelial cells. By this inhibitory action, transforming growth factor beta function to control immune and inflammatory response. Transforming growth factor regulates the differentiation and function of distinct of subset of T cell. As described earlier, transforming growth factor stimulates the development of peripheral FOXP3 Treg, best established in vitro. In addition, in combination with cytokine producing during innate immune response, such as interleukin-1, interleukin-6, transforming growth factor beta promotes development of Th17 subset of Th4 T cell by virtue of its ability to induce transcription factor roles. The ability of transforming growth factor to suppress immune and inflammatory response is part of generating Treg and to promote development of pro-inflammatory T helper 2 cells in the presence of other cytokine is an interesting example of how single cytokine can have diverse and sometimes opposing action depending on the context in which it is produced. Transforming growth factor beta can also inhibit development of T helper 1 and T helper 2 subsets. Transforming growth factor beta stimulates production of immunoglobulin A, IgA, antibodies by inducing B cell to switch to this isotype. IgA is major antibody isotope required for mucosal immunity. See in chapter 14 already. So transforming growth factor beta promotes also tissue repair after local immune and inflammatory reaction subside. This function is mediated mainly by ability of transforming growth factor beta to stimulate collagen synthesis and matrix modifying enzymes production by macrophage and the fibroblast and promoting angiogenesis. These cytokines may play a pathological role in disease in which fibrosis is an important component, such as pulmonary fibrosis and systemic sclerosis. Thereafter, let us talk of interleukin-10. Interleukin-10 is an inhibitor of activated macrophage and dendritic cell and is thus involved in control of innate immune reactions and cell-mediated immunity. It is the member of the family of heterodimeric cytokines that include interleukin-22 and others. The The interleukin-10 receptor belongs to the type 2 cytokine receptor family, similar to the receptor of interferons, and consists of the two chain which associate with the genus family kinase JAK and TYK2 and activate STAT3. Interleukin-10 is produced by many immune cell populations, including activated macrophages and dendritic cell, Treg, and T helper 1, T helper 1 and T helper 2 cells. Because it is both produced by and inhibit macrophage and dendritic cell, its function as negative feedback regulator. Interleukin-10 is also produced by some B lymphocytes, which have been shown to have immunosuppressive function and have been called regulatory B cells. The biologic effect of interleukin-10 results from its ability to inhibit many of the function of activated macrophage and dendritic cell. Interleukin-10 inhibits the production of interleukin 12 by activated dendritic cell and macrophage. Because interleukin-12 is critical stimulus of interferon gamma secretion, which play an important role in innate and adaptive cell-mediated immune reaction against intracellular microbes. Interleukin-10 suppresses all such reaction. In fact, interleukin-10 was first identified as a cytokine that inhibited interferons gamma production. Interleukin-10 inhibits the expression of co-stimulators and class 2 MHC molecule dendritic and macrophages. Because of this action, interleukin-10 inhibits T cell activation and terminates cell-mediated immune reaction. Infants who have homozygous loss of function mutation in interleukin-10 genes or in the genes for interleukin-10 receptor develop severe colitis before the age of 1 year. Knockout mouse lacking interleukin-1 in all cells or only T in Treg also develop colitis, probably as a result of uncontrolled activation of lymphocyte and macrophage reacting to enteric microbes. Because of these findings, it is thought that this cytokine especially is important for controlling inflammatory reaction in the mucosa tissue, particularly gastrointestinal tract. See in chapter 14. The Epstein-Barr virus contains a gene homologous to human interleukin-10, and the viral interleukin-10 has the same activity as natural cytokines. This raises the intriguing possibility that the virus has acquired the interleukin-10-like gene during its evolution, giving its ability to inhibit host immunity and thus survival advantage in its infected host. Roles of regulatory T cells in self-tolerance and autoimmunity. The elucidations of the genetic basis of IPEX syndrome and similar diseases in mice caused by mutation in FOXP genes described earlier is convincing proof of the importance of Treg in maintaining self-tolerance and homeostasis in immune system. Numerous attempts have been made to identify defect in development or function of Tregs in more common autoimmune and inflammatory disease in humans, such as intestinal bowel diseases, inflammatory bowel diseases, type 1 diabetes, and multiple sclerosis, as well as allergic disorders. Defect in Treg or resistance of effector cell to suppression of Treg may contribute to the pathogenesis of these diseases. However, it has proved difficult to definitively establish the role of Treg defect in those common diseases, in part because defining and quantifying functional Treg in humans is challenging. There is also potential for expanding Treg in culture and injecting them back into the patient to control the pathological immune response. Clinical trials of Treg transfer are ongoing in attempting to treat transplant rejection, graft versus host disease, and autoimmunity and other inflammatory disorder. In addition, clinical trials are underway to expand these cells in patients by administering the cytokine 2 in doses and forms that are preferentially bind to CD25 and thus activate Treg. In addition to the importance of controlling autoimmunity, Treg have been shown to serve other roles. Subpopulation of Tregs with unique transcriptional signature are present in many tissues and appear to perform a function that are especially benefit for those tissues. Treg that reside in skin, muscle, and other organs such as lung promote tissue repair and proliferation and differentiation of the stem cells, thus helping to restore tissue integrity after inflammatory reaction resolve. Adipose tissue, Treg control fat metabolism. Treg are also involved in maintaining fetal tolerance and preventing the rejection of fetus and play a role in preventing inflammatory response to the commensal microbes. Already seen in chapter 14. Deletion of T cells by apoptotic cell death. T lymphocytes that recognize self-antigen with a high affinity or repeatedly stimulated by antigen may die by apoptosis. There are two major pathways for apoptosis of apoptosis, both of which have been implicated in peripheral deletion of mature T cells. The mitochondrial or intrinsic pathway is regulated by BCL2 family of protein named after founding member BCL2, which was discovered as an oncogene in an B-cell lymphoma and shown to inhibit apoptosis. Some members of this family are pro-apoptotic and other are anti-apoptotic. The pathway is initiated when cytoplasmic protein of BCL2 family that belong to BH3 only subfamily, so called because they contain one domain that is homologous to third conserved domain of BCL2, are induced or activated as a result of growth factor deprivations or noxious stimulus, DNA damage, or certain types of receptor-mediated signal. This is e.g. strong signal derived by self-antigen in immature lymphocyte. BH3 only proteins are sensor of cell stress that bind to and influence this effector and regulator. In lymphocytes, the most important of these sensors is a protein called BIM. Activated BIM bind to two apoptotic effector proteins of BCL2 family called BAX and BAC, which they oligomerize and insert into the outer mitochondrial membrane, leading to increased mitochondrial permeability. Growth factors and other survival signals induce the expression of anti-apoptotic member of BCL2 family such as BCL2 and BCLXL, which function as an inhibitor of apoptotic by blocking BAX and BAC and thus maintaining the intact mitochondria. BH2 only protein also antagonize BCL2 and BCLX while when cells are deprived of survival signal, the mitochondria become leaky because of action of BHL2 only protein sensor and back effector and the relative deficient of anti-apoptotic protein such as BCL2 and BCLX2 and BCLXL. The result is that many mitochondrial components, including cytochrome C, leak out into the cytosol and activate cytosolic enzymes called caspases. Cytochrome C binds to the cytosolic protein called apex of 1, which then oligomerize and activate the pro-caspase 9, leading active caspase 9. Caspase 9 in turn cleave and thereby activate downstream caspase that induce nuclear DNA fragmentation and other targets that culminate in apoptotic death. So this is the pathway for apoptosis. And apoptosis is induced by mitochondrial and death receptor pathway described in the text, which culminate in the fragmentation of dead cell and phagocytosis of apoptotic body. So normally, this is the pathway. You have cell injury, deficiency of growth factor, survival signals, DNA damage. If this happens, then the mitochondrial intrinsic pathway is affected. Then BCL2 family effector, BAX and BAC, and BH3 only proteins. This one causes cytochrome C and other apoptotic proteins to be released. So regulator BCL2, BCL2 initiate the caspase space, and the death of receptor intrinsic death receptor extrinsic pathway are coming the first they are going in. So initiator caspase 8 execution of capses, then they can do the DNA fragment, then it die, liquid ligand of receptor on the phagocytosis taken, then it's there. In the death receptor or extrinsic pathway, cell surface receptor of the tumor necrosis factor receptor superfamily are engaged by their two TNF superfamily ligand. These receptors organize and activate cytoplasmic adapter proteins which assemble caspase 8, which cleaves itself when oligomerized and produce active caspase 8. The active caspase 8 cleaves downstream caspase, again resulting in apoptosis. In T cell, the most important death receptor is FAX, CD95, and its ligand is FAX ligand. FAX is the member of tumor necrosis factor receptor family, and FAX is homologous to tumor necrosis factor. In many cells type, caspase 8 cleaves and activates BH3 only protein called BID that bind to BAX and BAC and induce apoptotic via the mitochondrial pathway. Thus, mitochondrial pathway may serve to amplify death receptor signaling. Factor that determine the immunogenicity and tolerogenicity of protein antigen. This is table 15.2. Feature that further stimulate the immune response, feature that favor tolerance. Persistence, short-lived, eliminated by immune response. Prolonged, leading to the persistent antigens. Port of entry may again determine the antigenicity and tolerability. If it's subcutaneous, that favors stimulation of immune response. If it is also intradermal or it is absence from generative organ, that can favor the immune stimulation. But if it is intravenous, mucosa presence in generative organ, that can favor immunotolerance. Presence of adjuvant, antigenic, if the antigen with adjuvant, it can induce the stimulation. Antigen without adjuvant, low co-stimulation. Properties of antigen presenting cell, mature dendritic cell, they can cause immune activation. High level of co-stimulators can cause immune activation. Immature, resting dendritic cell, they can cause immunotolerance. In cells undergoing apoptosis, fragment of the nuclear cytoplasm breaks off in membrane-bound structure called apoptotic bodies. There are also biochemical changes in plasma membrane, including the exposure of lipids such as phosphatidylglycerin, which is normally on the inner surface of plasma membrane. These alterations are recognized by a receptor on phagocytes and apoptotic bodies, and cells are rapidly engulfed and eliminated without ever having elicited a host inflammatory response. This process is called the efferocytosis. Furthermore, phagocytosis of apoptotic cells may induce the production of anti-inflammatory mediator by macrophage. The best evidence for the involvement of two apoptotic pathway in eliminating the mature cells reactive lymphocyte is that genetic ablation of either in mice result in systemic autoimmunity, and the disease is more severe if both pathways are disabled. Mutation of FAXL or downstream caspase cause the human autoimmune disease called autoimmune lymphoproliferative syndrome. These two death pathways may function in different ways to maintain self-tolerance. T cells that recognize self-antigen in absence of co-stimulation may induce apoptosis by mitochondrial pathway. In normal immune response, the responding lymphocyte receives signal from T cell receptor, co-stimulators, and growth factor. These signals stimulate the expression of anti-apoptotic protein of BCL2 families, and thus prevent apoptosis and promote cell survival, the necessary prelude to proliferation. When T cells avidly recognize self-antigen, they may activate Beam, which trigger death by mitochondrial pathway, as described earlier. At the same time, because of the relative lack of co-stimulation and growth factor, and the anti-apoptotic member of BCL2 family remain at low level, and the action of effector backs and back are thus not counteracted. The Beam-dependent mitochondrial pathway of apoptotic is also involved in negative selection of self-reactive cell in thymus, described earlier, and in contraction phase decline of immune response after initiating antigen has been eliminated. Repeated stimulation of T cell result in co-expression of the death receptor FAX and its ligands FAX, and engagement of FAX trigger apoptotic death. When T cells are repeatedly activated, both FAX and FAS are expressed on the cell surface, and binding of the ligand to receptor activates cascade or caspase, which ultimately causes apoptotic death of the cell. The same pathway of apoptotic may be involved in elimination of self-reactive B lymphocytes, also in peripheral, discussed later. Factors that determine tolerogenicity of self-antigen. Studies with a variety of experimental models have shown that many features of protein antigen determine whether these antigen will induce T cell activation or tolerance. Self-antigen have several properties that make them tolerogenic. These antigens are expressed in generative lymphoid organs, where they are recognized by mature lymphocytes. In peripheral tissue, self-antigen engage antigen receptor in specific lymphocyte for prolonged period and without inflammation and innate immunity. The nature of dendritic cell that display antigen to T cell is an important determinant of subsequent response. Dendritic cells are resident in lymphoid organ and non-lymphoid tissue, may present self-antigen to T lymphocyte and maintain tolerance. Tissue dendritic cells are normally in resting immature state and express low level of co-stimulators. Some of them may traffic at low rate from epithelial to secondary lymphoid organs, even at steady state in the absence of infection and inflammation. Such antigen-presenting cells may be constantly presenting self-antigen without providing strong co-stimulation, and T cells that recognize these antigen become anergic or differentiate into Treg instead of effector and memory lymphocyte. By contrast, the dendritic cells that are activated by microbes and are principal antigen-presenting cell for initiating T response. As we will discuss later, local infection and inflammation may activate resident dendritic cell, leading to increased expression of co-stimulators, breakdown of tolerance, and autoimmune reaction against tissue antigen. There is a great interest in manipulating the properties of dendritic cell as a way of enhancing inhibiting immune response for therapeutic purpose. Our understanding of mechanisms that link the signal that T cells receive at the time of antigen recognition with the fate of what T cells remain incomplete. These concepts are based largely on the experimental model in which antigen are administered to mice or are produced by transgenes expressed in mice. One of the continuing challenges in this field is to define the mechanism by which various natural, normally expressed self-antigen induce tolerance, especially in humans. B lymphocyte tolerance. Tolerance in B lymphocyte is necessary for maintenance of unresponsive T-independent self-antigen, such as polysaccharide and lipids. B cell tolerance also play a role in preventing antibody response to protein antigen. Experimental studies have revealed multiple mechanisms by which encounter with self-antigen may abort B cell maturation and activation. Central B cell tolerance. During their maturation in bone marrow, B lymphocytes first express IgM as their antigen receptor and functionally immature at this stage. If these B lymphocytes recognize self-antigen in this bone marrow with high affinity, they either change their specificity or deleted. Receptor editing. If immature B cell recognize self-antigen that are present at high concentration in the bone marrow, and especially with antigen and especially if the antigen is displayed in multivariant form on cell surface, many antigen receptors on each B cells are cross-linked, thus delivering strong signal to cells. One consequence of such signaling that immature B cells reactivate their RAG1 and RAG2 genes and initiate new round of VG recombination in Ig kappa light chain gene locus. A VK segment upstream of already rearranged VKJK unite is joined to downstream the JK or J kappa. As a result, the previously rearranged V kappa J kappa exons in the self-reactive immature B cell is removed. A new Ig light chain is expressed, thus creating B cell receptor with new specificity. In this process is called receptor editing, and it is an important mechanism for eliminating self-reactivity from mature B cell repertoire. If the editing light chain rearrangement is non-productive, additional VK to JK rearrangement will be made in same locus, and if this fails, the process may proceed at kappa locus on other chromosomes. If that is non-productive rearrangement at lambda light chain lock may follow. A B cell expressing a lambda light chain is likely cell that has undergone receptor editing. It is estimated that among peripheral blood B cell in human, as many as 1/4 to 1/2 of all cells and majority of lambda expressing cells have undergone receptor editing during their maturation. Deletion. Strong recognition of self-antigen by mature transitional B cell in the bone marrow or soon after these cells emerge from the bone marrow may lead to apoptosis of B cell. This is presumably similar to apoptosis in T cell when high affinity antigen recognition in thymus trigger the mitochondrial pathway of apoptosis. But the mechanisms are not well defined. It is likely that self-reactive B cells first try to change their specificity and die if receptor editing fails. So this is the structure. We have the self-central tolerance in B cell. So A is immature B cell that recognize a self-antigen in the bone marrow with high avidity, that's multivariant array of antigen on cells, die by apoptosis or change the specificity of their antigen receptor editing, which involve only light chains but illustrated as change in the antigen binding region of the receptor. B is a weak recognition of self-antigen in the bone marrow may lead to the ANAG functional activation of B cells. So we have high avidity self-antigen recognition, self-antigen, then self-reactive B cell receptor editing, expression of new Ig light chain, then apoptosis will happen if editing is failing. If it happens that it is editing, then it works. Low avidity self-antigen recognition, self-antigen reduce the receptor expression signaling, it causes ANAG and sometime later death. This one is peripheral tolerance in B cells. B cells that encounter self-antigen in peripheral tissue become anergic or die by apoptosis. In some situation, recognition of self-antigen may trigger inhibitory receptor that prevent B cell activation. So this is what is happening. Self-antigen, we have it. A functional inactivation can happen caused to energy. If we meet self-antigen, some of them they can undergo apoptosis directly, or inhibitory receptor can be formed, causing regulation by an inhibitory receptor. Energy. If developing B cells recognize self-antigen weakly, e.g. if antigen is soluble and does not cross-link many antigen receptor, or if the B cell recognizes antigen with low affinity, the cell becomes functionally unresponsive, anergic, and exit the bone marrow in this unresponsive state. Anergy is due to downregulation of antigen receptor expression and block in antigen receptor signaling. Peripheral B cell tolerance. Mature B lymphocytes that recognize self-antigen in peripheral tissue in the absence of specific helper T cell may be rendered functionally unresponsive or die by apoptosis. Signal from T cells may be absent if these T cells are deleted or anergic in case of protein antigen, or if the self-antigen are non-protein antigen. Because self-antigen do not elicit innate immune response, B cell will also not be activated via complement receptor or pathogen recognition receptor. Thus, as in T cell, antigen recognition without additional stimuli results in tolerance. Peripheral tolerance mechanism also eliminate autoreactive B cells clones that may be generated as an unintended consequence of somatic mutation in the germinal center. Several mechanisms of peripheral tolerance in B cell have been described. Anage, some self-reactive B cells that are repeatedly stimulated by self-antigen become unresponsive to further activation. Immunologic B cells require higher than normal level of growth factor B cell activation factor, also called B lymphocyte stimulator, for survival, and they cannot compete with normal naive B cells for BAF, B cell activation factor. As a result, B cells that have encountered self-antigen have shortened life span and are eliminated more rapidly than cells that have not recognized the self-antigen. Deletion, B cells that bind with high avidity to self-antigen in the peripheral may also undergo apoptotic death by mitochondrial pathway. Deletion of B cell specific proteins antigen may occur outside the bone marrow at the transitional B cell stage found in the spleen and the blood. Transitional B cells are cells that are in this process of maturing in the peripheral into follicular B cells. A larger function of transitional B cells may be autoreactive, and most of them are deleted and do not mature into the follicular B cells. The mechanism of deletion of transitional B cells stage are not well defined. Somatic mutation of IgG genes during germinal center reaction may also lead to the emergence of self-reactive B cell. Some of these autoreactive B cells may be deleted by FACS pathway. Signaling by inhibitory receptor, B cells that recognize self-antigen may be prevented from responding to engagement by various inhibitory receptors. The function of this inhibitory receptor is not set threshold is to set a threshold for B cell activation, which allow response to foreign antigen, because this typically elicits a strong signal from a combination of B cell receptor, co-receptor, innate immune receptor, helper T cells for protein antigen, but does not allow response to self-antigen, which engage only the B receptor. This mechanism of peripheral tolerance was revealed by studies showing that mice with defect in tyrosine phosphatase, SHP1, the tyrosine kinase liens, and inhibitory receptors FC gamma RIIB and CD22 develop autoimmunity. CD22, also known as Siglec-2, bind to sialic acid that decorates sugar side chain of protein on the B cell's surface and dampen the B cell receptor signals. Immunoreceptor tyrosine-based inhibitory motif in the cytoplasmic tail of CD22 are phosphorylated by lin and this inhibitory receptor that recruit SHP1, thus attenuating B cell receptor signaling. How exactly CD4 contribute to B cell tolerance is not fully understood. Regulation of B cells by Treg. A specialized subset of Treg, called T follicular regulatory cells, enter the lymphoid follicles and contribute to the attenuation of T cell help to germinocenter B cell during antibody response. Defect in the regulatory T cells have also been associated with increased number of T follicular cells being generated outside the follicles at the boundary between B cell zone and T cell zones. Overall, Treg outside the follicles and T follicular cells in the germinocenter contribute together to limit the T follicular cells number and activity, and thus prevent a promiscuous B cell activation. Predictably, deficiency of Treg is associated with the autoantibody production. Tolerance to commensal microbes and other foreign antigen. Commensal microbes are abundant in the gut, skin, and other tissue, but do not elicit immune response despite being foreign. There are several reasons for this lack of immunogenicity. Many of these microbes cannot invade epithelial barrier and therefore may not be accessible to adaptable immune system. These microbes also induce and activate Tregs, which prevent development of effector and memory cell. Foreign antigen may be administered in the way that preferentially induce tolerance rather than immune response. In general, protein antigen administered with adjuvant favor immunity, whereas repeated doses of antigen administered without adjuvant tend to induce tolerance. The likely reason for this is that adjuvant stimulate innate immune response and the expression of co-stimulators on antigen-presenting cell, and in the absence of these second signals, T cells that recognize the antigen may recognize energy or die, may become energy and or die, or may differentiate into Treg. Many other features of antigen and how they are administered may influence the balance between immunity and tolerance. The oral administration of a protein antigen after lead to suppression of systemic humoral and cell-mediated immune response to immunization with same antigen. This phenomenon is called oral tolerance, we'll discuss in chapter 14. Therapeutic tolerance induction. Understanding how to induce tolerance by antigen administration is the key to development of antigen-specific tolerance as treatment strategy for immunological diseases. Numerous clinical studies are under the way to induce tolerance in autoimmune diseases and allergic in which the relevant antigen is known. The autoimmune disease include type 1 diabetes, and which is the major self-antigen insulin, MC, myelin basic protein, rheumatoid arthritis, and several strategies are being tried. Repeatedly administration of small doses of immunodominant peptide derived from self-antigen in the aqueous form without adjuvant. It is expected that antigen will be recognized in the absence of co-stimulation and will induce peripheral T cell energy and detection and deletion or preferentially activate Treg. But the cellular consequences of antigen administration remain unknown in these clinical studies. It is also unclear how effective this approach will be in inhibiting ongoing immune response in patients. This approach has been used for many years to treat allergies in a process called desensitization in peptide immunotherapy, seen in chapter 20. It is effective in subset of allergic patient, but how desensitization work is also not established. Administration of peptide derived from the self-antigen or coupled from the non-immunogenic substance such as nanoparticle and erythrocytes membrane. The idea is similar to the aqueous antigen administration described previously. Transfer or activation of Treg has been mentioned previously. Co-stimulation of blockage of refer to the blocking B7 molecule using the drug of CTLA and antibodies. It is likely this treatment inhibits immune response but does not induce long-lived tolerance. Despite the promise of these approaches, there has so far been little success in preventing immune response against autoantigen in patients with ongoing autoimmune disease. Defining mechanism for which different strategy may be working will require analysis of antigen-specific lymphocyte for which the technology is still developing. Next, we will discuss the mechanism of autoimmunity, and as of now, we stop here first.